All right. Good evening, everyone. Thank you for coming for your post-dinner education class opportunity tonight. Tonight we have Dr. Mike Sassner here. That last name may sound familiar to you. His dad is here, but Mike is actually I would say UNH. That's where my allegiance is. An Oyster River graduate. Went to Colby. Went to UConn. He was at the NIH for 5 years, and then he's been up at Jackson Lab for 25 years doing lots of different research, which he's going to get into. Um, and he'll say this, but as if you have questions through his talk, raise your hand. I'll come around with the mic, as you know, and we can ask those questions, so it can be more of a kind of a talk that way. So, um, without further ado, we'll make sure your mic is on and
Can you hear me
turn it
now?
over to you. Thanks, Mike.
Thank you. So as Jen said, I'm a proud Oyster River High School graduate, and I went to Colby for biology and chemistry, and then I studied neuroscience, and both at UConn and then postdoc at the National Health in Bethesda. And then I moved to the Jackson Lab 25 years ago. And so today I'm going to give you a little introduction to the Jackson Lab, talk a little bit about just general overview of Alzheimer's disease, and then I'll talk about some of the research we're doing now, a big project we have, and then a couple small projects if time allows. And I promise you I'll end with reasons for optimism. This is a hard subject to talk about. We've all dealt with Alzheimer's disease, and I've had Alzheimer's in my family, and it's a horrible disease. And I just want to let you know that there is progress and there is optimism. There are drugs in the clinic now, drugs in trials, and we're making progress. So I promise there'll be a light at the end of the tunnel at the end. So Jackson Lab, Bar Harbor, Maine. If you take the elevator over here, you see a big picture of Bar Harbor right next to the elevator, and actually there's a picture from MDI on the next to the workout room upstairs too, from Jordan Pond. We're an independent nonprofit biomedical research organization that leverages a unique combination of research, education, and resources. And this is the marketing speak, but it's real in this case. We discover precise genomic solutions for disease and empower the global biomedical community in its shared quest to improve health. What that means is we do our own basic research, and I'll talk about that today, but we're also empowering the whole community by generating resources, generating data, generating animal models, having courses, doing education. So we really make life better for everyone in terms of advancing research everywhere, not just in our labs. I have to share these pictures. This is Huguenot and Champlain Mountain. This is Park Loop Road if you've ever been to Acadia, and this is the Jackson Lab. I can walk from my office over here to the parking lot and go right up Huguenot and Champaign in about an hour and a half. It's a great place to work, great place to live. So JAX, we have about 60 different research labs, about 400 PhD and MD scientists. We have research centers, not just in Alzheimer's, but in aging, cancer, and computational biology. That's the research we do on our campus. We also give resources to, internationally, thousands of different organizations. By resources, I mean animal models. We have over 14,000 different types of animal models for different diseases. The COMP project, we knocked out each gene in the mouse so we can study what each gene does. We have genetically diverse models, and we have genetic databases. And then the third part is research, resource, and education. We give graduate degrees either with Tufts or University of Maine. We do postdoctoral research training. We have summer internships. We just started the Rue Institute in Portland for bioinformatics, and we have courses and conferences on our campus in Maine. And actually, when I was in Bethesda, we had two small kids. It's August. It was 95 degrees and humid. And I got a chance to go to course in Bar Harbor, Maine. I was like, ooh, yeah, I'll go. And I went up there, spent a week, and I said, if there's ever a job at Jackson Lab, I'm going to apply for it. I don't care what the job is. I'm going to move up there. My wife's from Waterville. I'm from New Hampshire, obviously. So it was great to get back to New England, and I feel really lucky to be here. It's a great place to live, and it was a great place to raise kids. This is the other view. You can see the Porcupine Islands, and this is Bar Harbor right here. So our funding is largely from federal grants for research. We also have grants from foundations. We have collaborations with pharma and biotech, and we get revenue from the distribution of mice that we develop, and then we also have a philanthropic arm. So I always like to thank the people funding this, the National Institute of Health, especially the National Institute of Aging, the Alzheimer's Association, and also this consortium for neuroimmune diseases. So please.
Go back to that last slide, please. Revenue from the distribution of mice.
So we create genetically modified mice. For example, the Alzheimer's models I'll talk about tonight. And we sell those to pharmaceutical companies, to biotechs, to academics. So each lab doesn't have to generate their own. It's basically a reagent, a tool you use in experiments, right? So if you're a pharmaceutical company trying to test a drug for Alzheimer's disease, we can sell you the mice to do that test instead of each company generating their own models. It's really important to have the same reagents used across different studies so we can compare across different sites. Exactly, standardization, yeah. So Alzheimer's disease is a type of dementia, but there are other types of dementia. So this is an umbrella term for loss of memory and other thinking abilities severe enough to interfere with daily life. Alzheimer's is the most common and one people think about a lot. But there's also vascular dementias, Lewy body dementias, frontal temporal dementias, or FTD, and other things like Huntington's and even Parkinson's has some aspects of this. What's important is that very few people just have one of these. You have 100 Alzheimer's patients, only 20 have just amyloid and tau pathology indicative of Alzheimer's. The other 80 have those things as well as Lewy body dementia or vascular or synuclein or Parkinson's. So all these things are very heterogeneous. It's not a pure disease. So of all dementias, half to 3/4 are Alzheimer's, a third or so, a fifth or third are vascular, and then Lewy body and frontotemporal dementia. So dementia is not one thing. There's lots of different things. And when we study this, when we develop drugs for these things, we have to understand what we're looking at and what we're studying. More than 6 million Americans are living with AD right now. By 2050, this will double. Alzheimer's kills more people than breast cancer and prostate cancer combined. There's huge amounts of money spent on, lost from these diseases and the effort lost and the care it takes. 11 million people provide unpaid care for people with AD, something like 18 billion hours of care, over $300 billion. All diseases are tough on the patients, but Alzheimer's is really tough on families, right? In many cases, it's a multiple years, it's a long process, it's a slow decline, and it's really hard to watch. And risk, if you're 45 now, there's a 1 in 5 chance for women, 1 in 10 chance of men getting it. So obviously there's a need for better understanding of disease and therapies that we can develop, and probably not to cure, but to prevent is the goal. You probably all know this, the warning signs, memory loss, challenges in executive function, planning and solving problems, having new difficulties with familiar tasks, confusion with time or space, trouble understanding visual or spatial relationships, losing words, misplacing things or getting lost, decreased or poor judgment, withdrawal from work, changes in mood and personality. Now, of course, all of us have some of these things, especially with age, right? All of us lose words once in a while. All of us lose things. My wife more than other people, evidently. But anyway, so there are, if you have one of these, there's no reason to be concerned. It's when you have lots of these together that there's concern. And it is a long process of development of Alzheimer's disease. These are put in standard size just for the figure, but these are not equal size. Preclinical phase of Alzheimer's disease can be decades, right? We know that some people start getting the pathology of Alzheimer's disease when they're 40s, okay? Even if they don't see the effect of it until their 80s. So you can start from preclinical AD where there's no symptoms, but we know there's biological changes. MCI or mild cognitive impairment, mild symptoms, they get more severe, where symptoms start to interfere with everyday activities, and then moving from cognitive impairment to dementia, where it interfere with many everyday activities, and then full-blown dementia. So there's lots and lots of risk factors, and we're really starting to understand what risk factors are important and how we can deal with those. Throughout the lifespan, I'll talk a lot about genetics moving forward, but I want to point out that things like diet, alcohol use, traumatic brain injury, concussion, smoking, metabolic issues like diabetes or dyslipidemia, hypertension, obesity, all these things can lead to a strong increase in your risk of Alzheimer's.
Like what are the risk factors of being a woman over a man? Twice as common.
Yes, but women live longer. Oh, sorry, yeah. The question was, is it true that women have twofold higher risk of getting Alzheimer's eventually than men do? Yeah, yeah. Whoops, sorry, I'm going the wrong way. Right here, yeah. So part of that is maybe, we're not sure, part of that is just women tend to live longer, right? But there are, we actually have a study right now with a group in University of Arizona where they think that the hormonal status going through menopause can have an effect on the energetic metabolism of your brain. Someone in the back's making faces at me. And therefore, your brain starts to use lipids instead of glucose, and that can lead to some changes in the brain that over time can lead to increased risk. So yeah, there are studies going on right now. We're actually looking at mice pre, peri, and postmenopausal and studying some of the biomarkers and seeing if we can correlate those things. So yeah, that's a good point. But point out too that there are protective factors throughout the lifespan. Education, physical activity, cognitive social activity, all these things can lead to a brain reserve. So even if you have a slight decline in your brain function, you're still competent and aware. And I wish I could tell you that there's some magic bullet here, but it's basically the things that your grandmother probably could have told you: eat healthy, don't drink too much, exercise. It's all common sense things. So from a neurobiologist perspective, what I care about is the pathology that causes the disease, right? Patients and doctors care about the symptoms. I care about how do we get to symptoms? How does a normal person develop Alzheimer's? So sometime in midlife, between your 40s, 60s, all of us start to deposit this protein called amyloid beta, which makes plaques in your brain. And this you can only see through postmortem staining, like is shown here, or with a radioactive ligand in the brain doing in vivo imaging. Once that starts, you start to have this immune cell in the brain called microglia activate. And you can see here, blue is the amyloid, and then green are all the microglia coming to this. And these are immune cells that try to get rid of the garbage, get rid of the toxic thing in your brain. Sometime after that, neurofibrillary tangles form. These are intracellular deposits from a protein called tau. And that leads to a breakdown in synapses, which is how neurons connect, and breakdown in neurons and neuron loss. And that leads to neural loss, atrophy, and then the symptoms that we're all too well aware of. So it's been known for a long time there's this progression of symptoms of pathology, starting with amyloid beta in cerebrospinal fluid, then precipitated A beta in your brain, then tau in cerebrospinal fluid, and then tau pathology. And then if you're high risk, meaning you had environmental risk, you would start to show dementia. And if you have low risk, meaning you led a healthy life, you'll be older before you started showing dementia. But this is the common pathway and the common progression of disease. We've known this for decades. The question is, can you diagnose it before the person dies?
Or is it just behavioral? I mean, behavioral stuff.
Yeah. That's a really, really important question. 10 years ago, I would say you can diagnose dementia, but you can't tell what type of dementia until you look post-mortem. Now we have these biomarkers, we have imaging, so you actually can determine, is this Alzheimer's disease? If you have sufficient, for example, if you have enough A beta in your brain, we can image that with this radioactive ligands and MRI. We can say, yes, this is amyloid. So yeah, we can do that now, which is a key point I'll get to, because until recently, even if we had a therapy for Alzheimer's disease, we didn't know how to give it to, right? If you can only diagnose after you're dead, it doesn't help you any, right? So now we can diagnose who has it, and we don't diagnose it as early as we'd like, but we can definitely define Alzheimer's disease based on biomarkers and in vivo imaging. So this is a nice story, and A precedes B, which precedes C, but that's correlation isn't causation. And a friend of mine wrote a book about studying Alzheimer's disease, and it's actually called "How Not to Study Alzheimer's Disease" because he was critical of because we've been doing this for 3 or 4 decades, and we don't have a drug. We're doing something wrong. So he kind of had this tongue-in-cheek. Well, orange here is gray hair. Well, gray hair precedes all these things, so maybe gray hair causes Alzheimer's, right? Just a kind of wise guy way of saying, just because B follows A doesn't mean that A causes B, right? I mean, so we need to look at our understanding of the biochemistry and the biology. And so the amyloid hypothesis, which we've been working on for decades, is that there's this protein called APP, or amyloid precursor protein. Protein gets cut in a specific way by these specific enzymes, secretases, and it forms this A beta monomer. A beta forms higher order structures, 2, 3, 4 together, make an oligomer. Then these plaques form, and then there's this magic arrow of, oh, plaques cause Alzheimer's. Well, there's probably 50 arrows between here and there, and this is what we don't understand. But this is the model we've been following for years and years. And there's a Aduhelm or aducanumab recently was approved, but we really don't have a cure. There have been hundreds of clinical trials that have essentially all failed. The cost for care is unsupportable. The prevalence is increasing. We're doing something wrong. We need to change the way we're doing things. So this is a complex slide, but I'll talk you through it. The bottom is showing frequency of a gene of genetic risk in the population. So here, these are the rare genes. These are common genes. The y-axis is your risk for Alzheimer's disease. So this is low risk, high risk, and causes Alzheimer's. The bottom of the bottom. So you can still hear me if I'm, yeah, okay. So we know this APP I talked about and the enzymes cut APP cause Alzheimer's disease. These are familiar early onset Alzheimer's disease. Genes expressed in neurons, and all the animal models we've had for the past decades are this type of Alzheimer's disease, familial early onset. Only 2% of patients have that. Okay, so we've been testing drugs in animal models trying to cure Alzheimer's disease. The models we've been using only represent 2% of the patients, right? Where maybe those are working in those 2%, but if you do a drug study in 100 people and only 2 respond, you say that the study failed, right? So this is just the past 2015 this is published. So now we understand, and I want to point out, 1 copy of APOE4 is relatively frequent and relatively moderate risk. 2 copies of APOE4 are moderate risk, but is also rare. This is the strongest genetic risk factor for Alzheimer's, for late onset Alzheimer's. So what's happening over here? This is where the action is, right? This is 98% of patients. These have late onset, which is the most common form of Alzheimer's disease. These are genes mainly expressed in microglia related to immune responses, right? So it's not the neurons we've been studying for the past decades. It's the microglia that have the genetic risk. So I spent all those years learning to be a neurobiologist. Suddenly I'm an immunologist, because this is where the action is. This is what's caused the genetic risk for Alzheimer's disease. So I call myself a neuroimmunologist. Save some face. But so we need to study these genes, these relatively common, relatively rare, relatively low risk genes, and what's causing changes in the microglia that lead to Alzheimer's. Now, microglia are immune cells that take care of garbage in your brain. So think of, you take 2 bags of garbage to the end of the driveway every week. The garbage truck comes, takes 2 bags every week. Homeostasis, you're garbage free. These are causing you to make 10 bags of garbage every week. Garbage truck is still taking 2 bags, so it builds up. That's a relatively rare form. As you age, the microglia get less effective. And if you have mutations, the microglia are less effective. So you're still making 2 bags of garbage a week, right? So you're not increasing the amount of production of A beta, but you're not dealing with it as well. That's partly due to genetics, partly due to age, right? So we need to understand what the microglia are doing so that we can understand the disease and then try to target and get therapies. So the new amyloid hypothesis is a cascade amyloid to different forms of amyloid to plaques to whatever. Microglia play a role in every step. We used to call microglia, we think of microglia as one thing. Then we had activated microglia versus quiescent microglia. Now we have disease-associated microglia, DAMs, barrier-associated microglia, BAMs, and there's many, many different forms of microglia. Really, all the action is studying these immune cells in the brain. So in 2015, the NIH had an AD summit in Bethesda and said, look, we got to do something differently. What we're doing isn't working. So they put out requests for proposals to fund a project to develop the next generation of animal models based on human data, establish a process for characterizing these animal models, make all the models available to everybody, including preclinical drug development, establish the features of these animal models in a translational way, and establish guidelines for preclinical testing. So before we had very similar models, modeling of small percent of the patients. Now we understand there's a lot of different types of patients we need to make models to model that 98%. So we, along with Indiana University and University of Pittsburgh, applied for this grant, won a $25 million grant for developing these animal models, and that's recently been renewed, so we're on the 7th year now of funding doing this. So just a quick tangent, why are we using mouse models to study Alzheimer's disease? And a mouse is not a human, a mouse brain is not a human brain. Mice don't live to be 80 years. But even though all models are wrong, some are useful. It's a matter of us being smart enough to use the models in the right way. So because the mice are mammalians, it's much more likely results can be translated to human population, as 98% of genes are conserved human to mouse. Obviously, mice live much shorter. We can generate a lot of mice quickly. We can generate 100 identical mice, give 50 a drug, give 50 a placebo, and see if that makes a difference, right? People, we're all different. We're all different genetic backgrounds, environmental backgrounds, diets, everything else. Those are really slow, expensive, painful experiments to do. If we can study 100 drugs in mice and then take 2 to the clinic, that's much more efficient than trying to put 100 different drugs testing in patients. We can genetically modify mice. We can knock a gene out. We can knock a gene in. We can humanize a gene. So really the goal is to make a model to serve as a vehicle for preclinical testing, so we can do this in mice. And so this is our focus, our goal. I don't want to say other things are not valuable, because we do some work in cell lines. We do some work in different mice models. And actually now there's a marmoset project funded by the NIH. These are monkeys that are think of them as big squirrels. They're very clever. You can teach them to do all kinds of cool things. They love strawberry milk, so you can bribe them with strawberry milk. They'll learn all kinds of puzzles. But they have Alzheimer's pathology like humans, and they live 10 to 12 years, so we can do it's still a long experiment. We can start to study these and understand pathology in these marmosets. So I love this quote from Sidney Brenner. Progress in science depends on new techniques, new discoveries, and new ideas in that order. People think of science as a bunch of smart people doing things. It's developing new techniques so that we can ask new questions and get new data. And that's really what I'm going to talk about, give examples of that moving forward. So this is called a Manhattan plot. It's supposed to look like skyscrapers. These are the human chromosomes across the bottom, and this log scale of genetic risk of Alzheimer's disease on the y-axis. You can see ApoE is the strongest genetic risk for late onset Alzheimer's. This is data from tens of thousands of humans where their whole genome has been sequenced. Okay, I started doing this 25, 30 years ago. I was sequencing one gene, right? It took me months. And that's all we could do. Now, I have a student, I say, go look up gene, APP gene, tell me the human sequence, tell me the mouse sequence, tell me how they're similar and where they're different. It takes them 5 minutes maybe. Okay, so we can sequence the whole 3 billion bases. We can sequence that in tens of thousands of people at once. And that's the only way we can get this type of data. So now we know genetic risk is there's lots of different genes that have genetic risk. These all interface with different I talked about A beta generation, but there's also immune function, lipid metabolism, blood-brain barrier. So all these things impact these different pathways that then lead to different types of disease. And so, think about curing cancer. We don't want a cure for cancer. We want a cure for liver cancer, a cure for brain cancer, et cetera, et cetera. Those are all different problems. Likewise, Alzheimer's is not one disease, it's many, many diseases. We're going to need different cures for different people at different stages of the disease. And of course, we always thought not just genetics, we need to include aging and environment. So the goal here is we know now we identify genetic risk. How do we get at the mechanisms? Because that's what we need to target pathways to try to cure Alzheimer's. And it's not just that, it's more complicated because actually these things interface with each other, right? A beta generation clearance relates to immune function. These things work on each other. This works on synaptic function. All these things then lead into this pathway at the bottom. So it's a bowl of spaghetti of interactions working on each other. So our goal with this NIH funding is take the human data represented by different colored people here with different types of disease, understand the proteomics and transcriptomics, what's happening with their gene and protein expression levels, their neuropathology, neuroimaging, and biomarkers. Take those genes, put them in mouse models, and then do the same experiments in the mouse models, getting the same assays so that we can correlate, does this red mouse look like the red person? And then we want to put that in preclinical testing. And this is one part of a bigger initiative funded by the NIH to try to cure Alzheimer's disease. So we started with this Manhattan plot. We prioritized genes. We picked genes based on lots of different categorization, how risky they were, how common they were, et cetera. So to generate late onset AD models, we introduced some strong risk variants. We humanized some of these genes. We developed combinations of these genes, APP and tau and other things. Then we introduced those risk variants that I showed you, put them in mouse models with different genetic backgrounds. And exposome is anything you're exposed to, obviously. Research mice are cared for very well. They're set humidity, set temperature, always have water, checked by a vet every day, fed this very healthy vegan diet, right? Well, I don't eat that. You probably don't either. And they also are very clean, so they have no immune challenge because they're very clean, right? When the power goes out at Jackson Lab in summertime and it's 90 degrees, my office gets hot. The mouse rooms have standard temperature, standard humidity, and mice are treated like gold, which is important for the research. You have to take care of your experimental models. So more just in the past year or two, we started using what we call a Western diet. So it's high fat, high sugar, which more closely stimulates what you and I are probably eating. And we're also looking at toxic metals, arsenic and lead in those things. We're seeing that genetic models that don't really mimic Alzheimer's very well, when you give them, for example, a high-fat diet, they start to more closely model Alzheimer's. We think a combination of aging, genetic risk, environmental risk is the way to go to get the best model to test drugs for human conditions. This is just a list to impress you of how many different mouse models we've made. Talk about it. So there is a lot of human data now. AMP-AD is Accelerating Medicines Project for AD, NIH funded. So what they have is hundreds of postmortem Alzheimer's disease brains and control, relatives sometimes or siblings or whatever, but they have control, quote unquote, normal brains. So they look at these brains, patients versus controls, and see which genes are changed, which genes are turned up, which genes are turned down with disease. And therefore, they can cluster these, and they know this group of 300 genes is turned up with Alzheimer's. We do the same thing in the mouse models, and we compare our mouse results to the human results. So blue here means correlated, red means anti-correlated, a box means it's statistically significant. So what we're seeing is that some of these mouse models have very similar groupings of genes turned up or down, similar to Alzheimer's disease. Right? So we think we're on the right track, at least for some of these biochemical pathways. We now have models. If you want to test a drug that's impacting the immune system, you can pick this mouse model off our shelf, order it, and test your drug, and we know that this is AD-like for this set of genes. And if your drug turns those things down, that's a good start toward therapy. This is at the transcript level. We also do this. Oh, I just want to point out that this is age-dependent, right? It's an age-dependent disease. It should be age-dependent. 4 months in this example, we don't see much correlation. 8 months, it's anti-correlated, but by 12 months, it's strongly correlated across here, right? So we're confident that we're going in the right direction because we're seeing age-dependent changes. And we're actually going out to 24 and 30 months now for some of these. I have to show this just because I love the name of it. It's called the spaghetti plot. These are different genes, how they are related to each other, and then we can group those in different categories. So this group of spaghetti right here is an immune response. This is a lipid metabolism response. This is vasculature. So we're seeing the right genes, the genes that are changed in Alzheimer's disease, we're seeing these genes change in the mouse models as well. So again, that gives us confidence we're in the right direction. And just showing here, just some data from some of these mouse models, we're seeing disease-like signatures with high-fat diet, HFD, and not without high-fat diet. So again, we're combining genetics and environment.
Mike, a question about the age of the mice.
Yeah.
Does that kind of correlate to middle age and older age?
Yeah, good question. And there's always trade-off. You don't want to wait 3 years for your experiment, but it's an aging phenotype. Yeah, so 2-year-old mouse, well, yeah, these we do 12 and 24 months. 12 is more like 35, and 24 month would be more like 65 or 70. So yeah, we are looking at those ranges. We're not going later, largely because the mice don't do that well after they get to be 2 or 3 years old. Is there another question there? But wait a minute. You get the microphone. Is it on?
I suspect you've been asked this recently, but in light of all the variables you're dealing with, does AI possibly be of any assistance in your analysis?
Oh. Yeah, that's a great question. The question was, with AI, would AI be useful for the analysis of this? And definitely in terms of the genetic data, we have lots of smart people working on that, but there's so much data. When I started, it was like the problem was making data. Now you have this fire hose of data coming at you and how you analyze it. So yeah, we are, that Rue Institute I mentioned, Northeastern, is a computational biology working on AI for this type of, for cancer and for Alzheimer's specifically. Yeah, exactly.
Yeah, in around 1985, Dr. Dennis Evans did the East Boston study about age, and he found that if you're 60 years old, your chances of getting Alzheimer's are 10%. And if you're 85, your chances are 50%. Does that study still hold? Has that been replicated by someone else?
Yeah, that's a hard question. I mean, every generation has different risks and different protective variables, right? So I'd like to think that the people who are, say, 40 years old right now are not eating McDonald's 3 meals a day, and they are more vascularly healthy, cardiovascularly healthy, and therefore the risk of Alzheimer's will go down naturally because we're living better. In fact, there was a story in the Boston Globe yesterday, the day before that, on the front page about this study of if you give a questionnaire of talk about your lifestyle habits, that can drive people to better lifestyle habits and decrease the risk of dementia because people are just more healthy in general. But yeah, there's lots of complications with that. The risk of Alzheimer's disease at 80, if you're dying of heart attack at 60, well, there's no risk, but it doesn't help you much, right? So that's a constantly moving target of the comparing different Alzheimer's disease risks with different generations.
The other thing I noticed on your risk chart early on, that a blow to the head was not included.
It definitely is. It wasn't on that chart, but there's no chart that's perfect. But yeah, that's definitely concussion and TBI is very much a risk factor. Unfortunately, we're getting a lot of studies from those things, largely from the military and in the NFL, I guess.
You talked about Indiana University.
Mhm.
I'm in that study. I wonder what you think of it, because I don't get any information of what they do for me. They want all kinds of information for me, but
So just to be clear, we have a project funded by the NIH to make mouse models to test drugs in. This is not a clinical study. The project I'm involved in is not doing any patient studies. That's a different project at Indiana, right? I'm on the basic research side, looking at doing research.
I think they seem to be looking at hereditary things more than anything. They ask me all the kinds of questions.
Oh yeah, yeah.
About the family.
Yeah, so that data would lead into one of those figures I showed you earlier about genetic risk in people. Right, that's not my study. I'm using that data that's published from those studies to generate the mouse models. So thank you for being part of that, because the field doesn't move forward without human subjects. So I want to point out too, we don't do a lot of cognitive studies because we think we want to be looking at the biochemical pathways and understanding the more direct link to disease. But this is something we just did in past year we're excited about. So this is percent correct in a mouse touchscreen assay, and these are the acquisition days. So you give them a task, again, you bribe them with strawberry milk or something like that, and they learn which light to go touch. And there's reversal training. You can do lots of different things, and the mice can really learn these things if they're motivated. So this is what a normal mouse looks like in red and black here. They start out, somewhere around 50% correct, and they get better over 3 weeks. Blue and red here are the late onset AD model with high-fat diet, both males and females. You see there's a significant difference here in the ability to learn a task between our controls and our disease models. The disease models on control diet are kind of in between somewhere, but definitely we're seeing severe cognitive deficits with a genetic model with the environmental risk of a high-fat diet. So we think this is a model, if you have a drug to cure Alzheimer's disease, these would be models. You could test your drug, and if we see this come back up again, you have very strong evidence that's something that should go into clinic and start testing on people. I emphasized early on that AD is a heterogeneous disease. Just very, very crudely, we're grouping it into inflammatory and non-inflammatory subtypes. And again, our mouse models, some are representing the A subtype, some are representing the B subtype. So we think we're getting different models to model different stratification of the patient population. And so it's a matter of not what's a drug for Alzheimer's, but it's which drug goes in which person. For time, I'm going to skip this. Just to point out that all of this stuff, we make all the data available publicly. We make the raw data available. We make summary target data available. We have an interactive interface. So if someone's in a pharma, they want to test a drug, they go in and say which model has changes in gene X. We can pop that out in 1 second, and they can pick the right model to do that. So it's all pushing things forward, not just for us, but for everyone. So there's this gene called Clotho, which is an aging gene. So I'm just going to talk about a couple short projects here. Again, this is funded by the National Institute of Aging. APOE4 carriers, and showed in red here, have a lifetime AD risk increasing as you get older. If you have APOE4, but you have this specific gene variant in the Clotho gene, you're protected. And the neutral is down here. So this is kind of a string to pull on, right? This is a handle. We can study this. If we can make a mouse model that has a difference in Alzheimer's disease risk, we'll understand what's causing that change. It's due to this interaction with this gene and this gene. We're looking at pathology biomarkers at 12, 18, 24 months of age in the different types of mice, whether they have the different versions of Clotho, the different version of ApoE. So this is one example of generating mouse models such that we can understand different types of disease, and even maybe protection from disease, right? We know which things cause disease. If we find something that prevents disease, that would give us something to target with a drug. So back to ApoE, I showed you this graph before. This graph was done 10 or 15 years ago. All the data was from European ancestry. All the work's done in Europe and North America. Most of the patients were white. All the data comes out like this. More recently, we've studied other patient populations. Europeans are here. Japanese have a much higher ApoE4 risk for Alzheimer's disease with this specific gene, whereas Africans have basically no risk from the ApoE4 gene, right? So suddenly, it's not a drug for Alzheimer's disease. It's a drug for different types of people with different genetic backgrounds. And luckily, we at JAX have a whole menagerie of different mouse models that come from different parts of the world. And we can show, so white here is the microglia I've been talking about, and blue are the cell bodies. These are just wild type. You can see they all have microglia. The bottom row is we introduce the APP or amyloid gene. This is the regularly used mouse. You can see the microglia kind of cluster around this nuclei like they should. In contrast, a different model called CAST is very, very aggressive. Microglia are very aggressive going after that amyloid plaque and clearing it. So CAST are more resistant to Alzheimer's pathology than the normal mouse. In contrast, microglia and WSB are lazy. They hardly even do anything. They don't migrate to the plaque, and they're very poor at clearing that. So the same genetic mutation on different mouse backgrounds has very different pathology. So this, again, gives us something, a string to pull on. This a handle we can look at. We can study the differences between these mouse models and understand what's causing the Alzheimer's disease. And finally, one more story. This got a lot of news. This is the New York Times a few summers ago. There's a family in Colombia, a large kindred, that have a mutation in that presenilin gene, which causes A-beta pathology. Everybody in that family with that gene gets the disease by 35, 40 years of age. Horrible, horrible condition. And of course, they don't know until they've had kids, and then it's passed on. People at Harvard Med School went down there, got to know the people, got to know the families, got a lot of blood. They were great about participating in these things, doing the genetics. And there's one woman who has the gene, so she will get the disease, except she didn't. So this is the PIB is the measure of amyloid burden. These are people with the disease. These are the controls. She's way up here. She's off the scale in terms of amyloid. Her head's full of amyloid. Never progressed to tau pathology. Never progressed to neurodegeneration. If you look at glucose metabolism as a measure of brain activity, she's right there with the normal people, whereas the other people, that mutation, are way down here. Okay, so again, this is an experiment of nature, a one-off, but it's fascinating. If we can understand what's happening with this point mutation in this one gene, we can understand possibly a way to target the gene pathway and maybe target the disease. So we have a grant from the Alzheimer's Association right now to make mouse models with these different variants. So we have 5 different cohorts. They have an amyloid driver, a tau driver, each of the versions, each of the flavors of ApoE, and then ApoE with and without that Christchurch mutation, which is protective. So if we can if these models show protection, now we have a tool. We can study these brains and understand what's happening in her brain versus all of her family, whereas she got the amyloid, but she didn't get the disease. And actually, people are now, this was just published recently, they're putting antibodies that mimic that ApoE Christchurch mutation as a potential therapy. So, also, I just love this. I said that 25 years ago we were sequencing one gene at a time. They found this mutation 2 years ago they went to a biobank and they have almost 500,000 samples. They can do the genetics on 500,000 people, right? Think about that. I mean, in terms of statistics, this is how you get things done. And it's all to the technology. So I promised you some reasons for optimism. So what I just said, genetic data, we can sequence all the genes in people in tens of thousands of patients. We can look at all their proteins, expression levels, all their metabolites, microbiome in their gut. We can now do gene expression in a cell-by-cell basis or brain region basis. And these are now happening not just in the European populations, but with a lot of diverse populations. We can genetically engineer models to be more realistic for testing disease. And we can make combination of late onset AD risk variants in these different backgrounds combined with environmental risk and aging. And therefore, a new understanding of disease mechanisms will enable new approaches for therapy development. So this is the drug development pipeline in 2023. So phase 1 is testing in humans. It's basically just a small number of people. If you take this drug, you get sick. Is it toxic? It passes that. You put a test on a small number of patients in phase 2, again, to assess safety and to understand how does a drug get in? You pee it out immediately? Does it get across the blood-brain barrier? That type of thing. Phase 3 is where the money really comes in, a large trial to test efficacy and safety. And then phase 4 is after drugs are already in the clinic and it's being used, following up on those patients. So outside circle is phase 1, inside circle is phase 2. This is phase 3. So now there are these drugs, largely antibodies to amyloid, which are in the clinic right now. And some of these are, so this is the good news. 10 years ago, this is a much less complex figure. But so we have drugs against amyloid, but it's exciting. We also have a lot of other targets we're hitting, right? So it's not just live or die by one target. We have, we're looking at vasculature, tau, synaptic plasticity, on and on and on. So there's lots of different targets, lots of different shots on goal. And, we may need different targets for different patients. We may need different targets for when we're treating people. So historically, especially in Alzheimer's disease, there's discovery phase, understanding a pathway, finding a target, optimizing the compound, hit that target, a lot of chemistry on the drug, preclinical development in a model, presumably, or in a cell line or whatever. Phase 1, phase 2, phase 3, and then FDA approval. This has always been the valley of death. It's easy to make compounds. It's easy to get hits in a cell line in a dish. It's hard to get across this pipeline. But due to the new technology and new funding, largely, we are getting across that now. We have a lot of phase 3 clinical trials, and these are sponsored not just by biotech and pharma, but academic medical centers. Some public-private partnerships are doing these. So there's lots of drugs in the clinic being tested right now, which is really exciting. 10 years ago, we didn't have that hope. Now we have lots of hope. So you've probably read about some of the anti-amyloid drugs. Lecanemab is one example. So this is months after. So give a drug at 0, go out to 18 months. And this is a clinical dementia rating. So blue is placebo, and you can see the decline in your cognitive test, basically, over 18 months. And this is a multi-point scale here. So the drugs are now not curing Alzheimer's disease, but they're slowing the rate of progression. So that's not ideal, but if that gives you another X number of years of cognitive health, that's a huge thing, right? If that gets you from one age to 5 years later, that's a huge thing. So we're really excited about that. There are issues here. There's some side effects. Depending on your genetics, you can be likely to get some type of swelling. A small number of people had brain swelling or even strokes. We now know that's risk is based on genetics, so we can take those people out of those studies, out of those treatment studies. So that is getting more safer. It's difficult because you need to do to administer the drug, you can't just give it a pill. You've got to infuse it, so that makes it hard for a lot of people to get. And you've probably read about the cost of some of these things are started out $60K a year, and they cut them down to $30K a year. So it takes a lot of time and effort and money to develop a drug, but if you price everyone on the market, it's kind of a waste of time. So that's a different discussion. So anyway, reasons for optimism in terms of therapeutic development. There are drugs in the clinic now. We now know how to match a drug to a person, how to understand the risk factors for each drug. We have biomarkers and blood tests to understand how to treat earlier, who should get the drug, when they should get it, and matching it to the person. Right now, there's anti-amyloid antibodies in the clinic being combined with anti-tau antibodies. So we know the anti-amyloid antibodies work to slow the rate of progression. Maybe adding a tau, maybe doing it 10 years earlier would get to be curative. And it's really fun for me as a scientist. There's lots of diverse approaches going on now. There are vaccines in the clinic. There's viral delivery of some of these protective proteins. We're actually using CRISPR to genetically engineer neurons, plasma replacement of factors like Klotho. So there's lots and lots of activity in this space. We give a course teaching all this stuff because we want to share the information. Course is held in this mansion that someone gave to the Jackson Lab a long time ago, right on the waterfront. So we spend the morning in the conference room up here doing lectures. They spend the afternoon in the lab learning how to drug mice or test mice or whatever. And then at night we just sit around and talk about ethical issues and training issues and career issues. So it's really fun to pass on what we're learning. Large number of people, need to thank all those people. These are just people at JAX who are now studying Alzheimer's disease. Purple shirts are for The Longest Day. It's a fundraiser for the Alzheimer's Association every longest day in June. Every year we meet on the top of Cadillac at sunrise, which Cadillac Mountain is the highest, most eastern place on the eastern seaboard. The sunrise is something like 4:45 a.m. Some of the students hike up in the dark with headlamps. I don't. I meet them there and I'll walk down with them. But then we spend the day hiking Acadia, or biking or kayaking or going around town trying to raise awareness for the Alzheimer's Association and fundraising for them. And a couple years ago, we actually did the shortest day, so December 21st. We went to Seal Harbor Beach. It was one of those classic Maine days where it's like 33 degrees and raining. So the ocean was the same as the outside, so it didn't really matter. So there's talk about doing that again this year, but we'll see who shows up. Anyway, thanks for your attention. I'm happy to take more questions. Oh, but that was the cover of the Globe the other day. Mass General is doing these tests to assess risk of dementia. Really interesting.
So did one of your charts say that, looking at the Japanese versus some of the other groups, that they had a higher risk? Because we think of them as a low-fat or certainly not a meat-eating diet. Did that surprise you?
No, that was showing the genetic risk for that one specific mutation.
Oh, okay.
Varies by your ethnic background.
Not Alzheimer's in general. Or,
yeah.
Okay.
Yeah. I mean, we have seen that as populations get more westernized, if you will, and eat more McDonald's, they tend to get less healthy. That's where you're going.
Your presentation is on Alzheimer's, and my question is any of this research translatable for helping with the other kinds of dementia, like Lewy body?
Yeah.
Or are there people in those areas like you who are doing the research?
I've actually worked with the Michael J. Fox Foundation for Parkinson's Research, generating mouse models for Parkinson's in similar ways. So basically, the trigger for these diseases are different, but the downstream effects, the neuroinflammation is the same in all these diseases: Huntington's, Parkinson's, Lewy body disease, Alzheimer's. So the microglia effects are the same across all these diseases. And some of the resources we're generating, we have tools, we have mouse models that have green fluorescent protein when microglia light up, for example. So those are useful for Parkinson's or Lewy body or any neurodegenerative disease.
I have a question. I understand and appreciate all Jax is doing for us and the world. What are the pharmaceuticals company doing, and how do you all fit together?
First, let me say I'm not an expert on pharmaceuticals, but we work closely with this company called Denali Therapeutics in South San Francisco. They've been great in terms of generating resources that are available. They call it pre-competitive, like making a mouse model, making an assay. It's not their compound they're using to cure Alzheimer's disease. It's a resource that everyone can use, and they've been good about sharing those things. So some of these farmers are very good. Historically, they've been horrible about sharing data. I've tried to get just negative data, right? Or placebo data, right? You do a study and just show us the data you used in your untreated people, and they still won't share that type of stuff. So, but they are putting tons of money into it. And as we get closer and closer to these things, they are certainly putting huge amounts of resources into it. You know, frankly, as investments and there's lots of IP, but it's much better than it was a few years ago. Before people could patent literally the human gene or the human mutation, which kind of froze research in certain areas. There was a famous case with BRCA2 where one company owned that gene. So if you want to test for breast cancer, you couldn't do it. Only one company could do it. So they changed the rules, so the laws. So you can't patent a product of nature now. So those things have gone away. But they're investing tons of money, is the short answer. Do you want to follow up? I can repeat it if you want. Oh. So the question is about competition between pharmaceutical companies and do they trust each other? I trust what's published because it's peer reviewed, right? And presumably peer reviewed data can be reproduced and other people can show. So if you publish a paper and I'm not sure about it, I will get the same mouse and the same antibody and the same assay and do the experiment over again and say, yes, this works out. Okay, in the vast majority of cases, that's true. I hesitated a minute there because there was a story in one of the journals just a couple weeks ago about a famous Alzheimer's researcher who, the results looked too good, and they went and studied, and some of his postdocs were like, yeah, we were pressured to get the right answer in some of these things. So, but the scientific endeavor is set up so that if your work can't be repeated, then it's useless, and there's no gain in faking results because it can't be reproduced, and no farmer's going to want to buy your IP because it's useless. So, I said trust would verify, right? That's the famous, right? That's where we are.
I have a question about environmental factors.
Mhm.
You were talking about, food and stuff. What about pollution?
Yeah, great question. One of our colleagues at Indiana is typing in ozone smog into mouse cages. And again, just like with heavy metals, just like with high-fat diet, they're seeing, and we don't know why yet, but we're definitely seeing stronger evidence of disease based on pollution. And there's been epidemiological studies for a long time showing if you live near a highway or in a congested city, you're more likely to get certain diseases. Not just asthma and things like that, but also neurodegenerative disease. So, yeah, that definitely is a part of it. And that's why it's so hard to study humans, right? Because we all have different diets, different environmental exposures, different genetics. That's why having animal models where they're genetically the same and can be treated the same is a way to move the field forward. Yeah. The question was, what's showing up in autopsies of centenarians? I was at a conference this summer with someone who runs the centenarian study in, let's say, Finland or Belgium. And she had some really cool data. So there are, these risk factors. I can name 5 genetic risk factors. In her group of, I forget, 200 centenarians, healthy centenarians. These people are fine at 100 years of age, cognitively fine. Every one of those people had the protective variant in these 5 key genes. That's not a coincidence, right? So, and that's rare. I mean, the rare variants may or may not be, the protective variants may or may not be rare, but the combination of all those 5 is going to be rare. And so for her to find, hit that combination of 5 lottery tickets 200 times in a row to get those people, that's not a coincidence. So, that's kind of sad news because genetics play a big role. But unless you were dealt the right hand. But certainly we know that, as I said, the environmental issues can be as strong an effect. But yeah, it's always best to have the right parents.
Are there certain parts of the country where Alzheimer's is much more active than other parts of the country?
I'd say there's socioeconomic groups that have higher risk. I mean, it's not geographical, it's socioeconomic. As we said, higher rates of education, higher rates of healthy lifestyles, are less likely to have cognitive or any type of dementia. Whereas poor neighborhoods with pollution and less access to healthy food and those things, there's certainly higher rates of cognitive decline, of dementias in those. Of course, there's also the confounding factor of low socioeconomic neighborhoods don't have medical facilities and clinics, so they don't get diagnosed as well either. So it's hard to make a concrete conclusion from those studies because you're comparing apples to oranges in every case.
Can you say that there's less Alzheimer's in a state like Arizona?
Well, old people move to Arizona, right? So, so there's more Alzheimer's because there's more people 80 years old in Arizona, right? Just by demographics. So like I said, there's lots of confounding factors.
Can you relate it to weather? In very cold climates, is there less Alzheimer's or more Alzheimer's?
I haven't seen studies on that. Yeah, I wouldn't want to guess. Coming from Maine.
There's been a lot of information about the microbiome and its connection to the brain. Have you found that there's more incidence with people with digestive disorders or digestive diseases?
Well, so it's 2 things. One is the metabolic health, which we'll separate for now. Definitely, there are studies showing that microbiome can have effects strongly in Parkinson's, but also in Alzheimer's disease. We actually ran some of those studies, and it's really hard to run because there's so much noise in the system. The noise is bigger than the signal in a lot of cases. Mice, you co-housed mice, they're eating each other's poop. It's really hard to run those studies. But there have been studies showing that specifically with the ApoE alleles, the microbiome they have can vary and can lead to more risk. So, but we don't know what's good and bad. We don't know what's driving these things. We're just seeing a correlation. So, but there is a lot of work going on in Parkinson's right now. Evidence, much stronger evidence in Parkinson's field that the GI effects can circle back to the brain.
Any other questions? No? All right. I know Mike had in one of the slides kind of 3 different websites to get resources or information or raw data from. I'll make sure to grab those from him, and I'll put them in the Wellness Wednesday.
Yeah, the Alzheimer's Association has great resources for scientists, for lay audiences. It's a really good organization. I'd recommend looking at that site if you're.
All right. Thanks, Mike.
Thank you.